Hello, people. Hello, friends. I've gathered you together today to discuss something of vital importance to all of us: developmental biology. So I'm a little bit worried about my computer. It's at about 50% now, 56%. It may die while I'm lecturing. If that happens, don't run away. I've got my wife's computer here, which isn't quite working either. But if it does die, I'll try and get on with her thing. But I'm not sure how long this lecture is going to go anyway. Half an hour might cover it. Okay, how many people we got? 20. 20 people. Bimalak. Elijah never misses. Neither does Esha Gupta or Michelle Simmons or Keela Simon. You guys are the stalwarts. You're here. You're here. Stefan's here, whether we like it or not. He's here. Nothing personal, Stefan. It's just, you know. Oh, I should stop teasing people. I should, but I can't help it. Can't help it. Oh, now we can start Abdusa boards here. And Dyson checking in from Azerbaijan. Dun, 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 okay, gonna pull up my PowerPoint, do a quick review of what we talked about last time. Did we talk about this? I don't think so. I think this is today. Blah, 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 blah. Okay, yes. Here's where we were last time. I'm going to put my notes for myself to follow along. 6. Okay, so last time we were talking about how gradients can be used to make an axis or axes, if you want to talk about it in plural. And so we ended talking about how the Speeman organizer creates this gradient of BMP4 activity. And it's doing that because it is not producing BMP. Actually, it does produce BMP4. But what it's doing is releasing these blockers of BMP4. So they're going to be high in concentration here and low in concentration here, which means that BMP4 activity is able to be expressed down here. And as you get closer and closer to the organizer, the less expression you can get from that, the less expression you get of the BMP4 because the blockers are getting higher in concentration. And here we can see it again. So I'll talk about this later today in terms of the neural specification included. And here, anterior specification of that endoderm there. Ba, 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 ba. So sometimes it's hard to remember that we've got a three-dimensional structure here when all the pictures look like this. But remember, you've got your blastocele here. This is still the blastula. And BMP4 is being released here from the endoderm. So it's pretty much everywhere around the embryo. It's like it's everywhere in the embryo until the Speeman organizer starts to kick out these blockers. And these blockers are diffusing away, so they're blocking in that direction. They're also blocking in this direction up here and this direction down here. Okay, so it's kind of like enveloping, kind of like if you dripped melted ice cream on top of a cue ball, you know, and start at the top and go around like that. I don't know where that metaphor came from, but there it is. And we also talked about this, another aspect of the blocker that is producing more of a gradient, and this is chordin, which is being produced in the Speeman organizer. And as it moves this way, it picks up BMP4, shuttles it down to the other side, And when it gets down here, the chordin is broken down by ADMP, and then it releases BMP here, which can then start moving this way again. So you've got that, you start to get a gradient of BMP moving in this direction, as well as blockers coming from this direction. So you get a nice gradient there. And that's going to be our dorsal ventral axis, because dorsal structures will form when BMP activity is totally knocked out. So that is our dorsal ventral axis. So today we're going to start talking about the anterior posterior axis and how this gets set up. This is also being set up by the Spemann organizer. And that's because of mainly effects on BMP, but also effects on Wnt. So here are some of those early experiments we talked about. These are like 1930, 1940s, before they had any molecular tools. They were playing around with embryos. So here's an example where you take the Spemann organizer out of an early gastrula, you stick it onto the other side of a different gastrula. So here's your blastoportal lip, here's your blastoportal lip, take it out here, stick it on the other side, you get two heads, right? So that we've seen before, that's not a big deal. What they're showing here now is if you wait till the gastrula has moved along a little further, and then you take that out and you put it in the same place on another blastula, you don't get a head anymore. You get a tail. You get a kind of trunk and tail being laid out there. And the difference between head and tail is anterior posterior. So what they're showing here is that anterior posterior axis is all contained within the Spemann organizer at the early gastrula stage. And then it's going to be laid out as gastrulation happens, because it's kind of like you've got that whole AP axis and it's like all crunched together like this, and it's going to spread out as it gastrulates. So we can see that better here. Okay, so I just want to make sure I'm on the right slide here for what I'm talking about. 48. Okay, so 47, 48. I don't have it listed in my notes, but anyway, so here's your Spemann organizer, and like I say, the whole AP axis is here. This is dorsal side, this is ventral side. Now, when gastrulation starts to happen, the very first cells here, the leading edge cells, are anterior endoderm. So these end up being like the bottom side of the face. So anterior is head, endoderm is the bottom part. So that ends up being that. I'm sorry, I'm just rearranging my real estate here so you can see this better. Okay, so the organizer itself is a mix of dorsal endoderm and mesoderm. Okay, dorsal endoderm, which is this here, and mesoderm. So we've got, this is all mesoderm here, and it's all scrunched up. So gastrulation then is going to roll this out. As the leading cells go, they're going to be precordial plate mesoderm. So precordial plate, we talked about before, is basically the head mesoderm. And then the brown underneath are called deep cells here. Oh, I'm sorry. The orange is the anterior endoderm. So again, the lower part of the gut that comes in this way. And then here is precordial plate mesoderm, which is the head. And then here is your notochord, which is going to make the spine. So as that then stretches out like this, now you've got your anterior posterior axis. So you can see how, or why I say you've got the whole AP axis scrunched up here in that Spemann organizer, because everything is here and then it peels out this way during gastrulation. And as it does, it leaves these things behind in different positions. And now you've got anterior endoderm here. You've got precordial plate mesoderm head. And then you've got notochord here. And what's going to happen next is this notochord is going to turn this ectoderm into spine. You're going to get neurulation that way. And then this is going to turn the anterior neural plate into brain. So we've spread that whole axis out that way. Okay, so here, these are some of the transcription factors that are going to specify this orange part is vegetal endoderm. Now, don't worry about these now. I may talk about them later with some of the other models. Right now, just imagine this as ventral endoderm. And the dorsal parts in red are expressing the transcription factor NOT, N-O-T. And I'm pretty sure that NOT comes from the fact that when this is going to be mesoderm or notochord. So when NOT gets expressed, those tissues, those mesodermal tissues become notochord. So you're seeing NOT expression here. Okay, so why is this happening? Why is this happening to me? So we've got two different gradients going on here. I'm not sure if I talked about this last time. I don't know. But one is your BMP4, your BMP gradient, which I talked about just now, and the other is a Wnt gradient. Wnt is really high in one area and low in the other. So right here at the blastula stage, we have talked about this, where the sperm came in here, you get your cortical rotation, so you get Wnt signaling over here. So Wnt signaling is beta-catenin. So there were, I think, 4 different things in this area that were dorsalizing, and they were all increasing beta-catenin. So your Wnt signaling is really high over here and low over here. And because you've got high beta-catenin here, along with veg-T, you get production of the nucoop, which is produced by CMWA, the transcription factor CMWA. CMWA then increases the, I'm just reviewing for you as I go through this, just so you know, you've heard it several times. So CMWA then induces the cells above it right here into becoming the Spearman organizer by upregulating goosecoid. And then that's going to produce and release our BMP antagonists as well as Wnt antagonists. So at this point, you've got this Wnt gradient from low to high here, and now you're going to produce Wnt antagonists over here in the Spearman organizer. So when you get into the after the gastrulation stage, when you get a neurula, now you've got very, very low, really no Wnt at the head and high Wnt at the tail. So you can imagine here, you've got high Wnt, but then when the tissues from here go forward, they end up with no Wnt, and Wnt still stays high back here. Okay, so we want to talk about why that is. What do we got here? So the Spearman is producing these antagonists. I could also add here Cerberus, right? Frisbee, Dickhoff, and Cerberus. And then the Wnt gradient during gastrulation, again, is because those leading cells are releasing a lot of Cerberus, which is making really high Wnt blocking activity at the leading edge. So that becomes anterior. And then the trailing ends have very low antagonists, so they end up being posterior. This chapter is a little bit repetitive. It kind of says the same things over and over again, but I think it bears repeating. So here again is your Wnt gradient after neurulation, or after gastrulation anyway. And you can see how that lays out your AP axis. Very low Wnt over here becomes anterior. Higher Wnt over here becomes posterior. Okay. So the Spearman then is setting up 2 different gradients. One is the BMP gradient from high down the bottom to low up top, and that's ventral to dorsal, okay? BMP will specify your ventral structures. So you've got this gradient of BMP from the bottom to the top, which is your dorsal ventral. And then you've got this other axis of Wnt expression in this direction, which is going to be a low Wnt, high Wnt, and that's your AP axis. And you can see it here again another way if this helps you. I'm not going to dwell on this one though, because it's the same thing, just looking at it differently. Okay, neural induction. Let us talk about neural induction. I'd like to induce a little neural activity in you. That was dumb. Okay, so again, early experiments. They don't have any molecular tools or really knowledge at this point. This is the 30s and 40s, so they want to figure out, where does neural specification come from? Did it start in the egg, or does it get induced later? So here's one experiment to show that. So you take one embryo that's at the gastrula stage, and you take this little, actually this is blastula, but it's just pre-gastrulating. And you take some Spearman organizer there, you stick it into the other end of a different embryo, and that piece of what would become neural tissue when put at this end of the embryo becomes epidermis. Okay, so you basically you pull this out and you label it pink, you stick it in at the ventral end of the blastula, and it ends up as epidermis. Now, if you take a piece of what's prospective epidermis, what will become epidermis out of one blastula, and you put it into the dorsal side of a different blastula, now it becomes neural tissue. Okay, so what's this telling us? This is telling us this is the animal cap here, which is all ectoderm. And if you take it out and culture it by itself, it all becomes epidermis. But if you take a piece of it out and put it over near the organizer, the Spearman organizer, it becomes neural. If you take out a piece that would become neural and you put it at the other end, away from the Spearman organizer, it becomes epidermis. So clearly, the organizer is causing this induction of neural tissue from the ectoderm. So that's step one to figure out what's going on here. And then here's what is this, in situ hybridization we talked about, where you take a piece of DNA and you label it somehow and you find out where the expression is. So that's one thing you can do with that kind of mechanism, use a normal embryo and find out where this thing's being expressed. Another really important use for this technique is finding out what the tissue is. Because, you take this early embryo and you change something, it goes from looking like a weird blob to a different weird blob. Sometimes just anatomically, it's hard to see what actually happened. So here, they're taking two different in situ probes, SOX2 and cytokeratin, and they're using them to differentiate between two different tissues, epidermis versus neural tissue. So if you see expression of SOX2, that means it's neural tissue. You see expression of cytokeratin, that means it's epidermal tissue. So what they're doing in this experiment is they're injecting antisense RNA against BMP into both cells at the 2-cell stage, and then they're letting it develop. So antisense RNA we've talked about before too. It's basically the complementary strand to the translated strand, and so you inject that, and it binds to all the BMP messenger RNA in the 2-cell stage, and double-stranded RNA gets cut up by enzymes in there because it's not supposed to be there. So that gets destroyed. So basically, at the early embryo stage, you're getting rid of all BMP. What happens with no BMP? What would be neural tissue? This is normal. And then you've got skin on the outside. Okay. So normally you've got neural tissue in the middle, which is the blue here, and epidermal tissue on the outside. Okay, this is the gastrula. Neural tube has not closed up yet. Now you get rid of all BMP, all you get is neural tissue, okay? All the ectoderm turned blue, and now you see no epidermal tissue, no cytokeratin being expressed, which means no epidermal tissue. So clearly BMP is really important for specifying neural tissue, because if you knock it out, you don't get any. So here's your BMP blocking diffusing things, chordin, noggin, follistatin, XNR3. So they're starting at the Spielman, and we've talked about this giving us mesoderm, dorsal and ventral mesoderm, but also it's going up this way, and it's blocking the effect of BMP so that this end of the blastula will be neuroectoderm in the end, okay? It's not right now, but it will be because this BMP blockers are coming up this far and blocking that. You're also getting dorsal endoderm down here because the blocking factors going in that direction. So this is BMP gradient here, and it gets blocked there and there. So that's where you get the neural. So we know BMP is important for neural induction, because if we totally block it, it does not get formed. Another thing about this is if you knock out one of them, hang on, let me make sure I'm getting this right before I say it wrong. Okay, so if you all three blockers. Okay, so we've got three different blockers here, chordin, noggin, and follistatin. And what they found, so what they did in the previous one was knocked out BMP, okay? So no BMP. Here, in another series of experiments, they knock out chordin to find out what happens. Not a lot happens. They knock out noggin, not a lot. Very underwhelming. You need to knock out all three of them in order to knock BMP signaling down enough that you will see a ventralized embryo, because you no longer have that. So that's just another piece of info there, which is that you need to knock out all three blockers in order to what am I trying to say? There's a lot of redundancy here in that you can knock out one and you don't see much of an effect, but once you knock them all out, then you don't get that neural ectoderm anymore, because you have to knock out all of the BMP activity in order to get that. Okay, so that's the importance of BMP. Another important signal here for the same reason is FGF. If you remember from previous lectures, here in this mesoderm, you get brachyury coming up. It's a transcription factor. Brachyury induces the expression of FGF, which induces the expression of brachyury. So we've got that positive feedback loop. But also FGF is important for blocking the BMP4 signal. And it's going to be expressed mainly at this end over here. And it does three things to block that FGF signal. It blocks the production of BMP. So it's blocking the transcription and translation of BMP. So there's less BMP wherever there's FGF. It increases the expression of chordin and noggin, and those are blocking BMP. And it also phosphorylates MAP kinase, which is activated by that FGF pathway, will phosphorylate SMAD1. And SMAD1 is necessary for BMP4, BMP signal transduction. Again, we talked about that a few lectures ago, where BMP activates SMADs 1, 5, and 8. And they all bind to SMAD4. And your XNRs are binding to SMADs 2 and 3, which also binds to SMAD4. So anyway, FGF is going to further block BMP by blocking BMP signaling. So it's blocking BMP production, and blocking BMP activity by putting the antagonist out, and blocking the signaling by getting rid of SMAD1. So FGF is also decreasing BMP, and that's allowing neural induction to happen as well. Okay, so that's important things for your neural induction is number one, get rid of BMP4. With that around, you're not going to get anything neural. So there's a lot of mechanisms there to knock its activity down. Okay, mesoderm. So mesoderm is also important in patterning of the neural tissue. So the pattern of the neural tissue is basically brain, spinal cord. You've got the top of the spinal cord down to the bottom of the spinal cord. So that's your AP patterning. So the mesoderm that comes rolling off the during gastrulation is going to cause that patterning to happen in the neurula. So mesoderm is inducing neural tissue from epidermal tissue, and it's also patterning it from A to P, anterior, posterior. So here's some of those early experiments that show this. In this one, they take a gastrula and they pull the neural tissue out and grab some of the mesoderm that's underneath it in the anterior part of that gastrula. And now they're going to a blastula here, and they're putting that anterior mesoderm in the ventral side of the blastula, and they end up getting two axes, and they actually get a brain over here in this head. So you've got a brain here and a brain here. So this is probably then what they're concluding here is this anterior mesoderm is producing anterior neural structures, right? It's doing some anterior patterning on the neural tissue. Here's the opposite experiment. So they're going to take some posterior mesoderm from the gastrula, put it in the same ventral region here of this blastula, and you get two trunks. So you don't get anterior structures, you get a posterior structure. And again, it's got a spinal cord showing that we're inducing ectoderm into neural ectoderm with this posterior mesoderm. Okay, so mesoderm's got some kind of a pattern to it, and you can transfer that from one embryo to another and get the same patterning. Okay, hang on one sec. I don't go too fast. How is this happening? How is this happening? It's happening because your anterior mesoderm, your anterior mesoderm is producing Wnt antagonists, okay? Whereas the posterior mesoderm is not. So our Wnt antagonists, if we recall, are Fris-B, which is binding Wnt extracellularly, Cerberus. I keep getting stuck. There's this series of comic books that came out in I think the late 80s, early 90s called Cerberus, but I always pronounced it wrong. I always pronounced it Cerebrus. I don't know why, but I did. And now we still get stuck with that word somehow. But anyway, that's neither here nor there. Good series of cartoons though, if you like graphic novels. See how you learn from me just from being here? It's amazing. It's amazing. What the hell was I talking about? Okay. So Fris-B, Cerberus, and the third one, which I was stalling for time to try and remember, is our old friend Dickoff. Okay, so the anterior mesoderm is releasing Cerberus, Dickoff, and Fris-B. Fris-B is an antagonist. It binds the hormone extracellularly. Dickoff is also an antagonist. I forget exactly how it works. Cerberus is going to be an antagonist of Wnt and BMPs. So It's a goodie. All of the dorsal mesoderm releases BMP inhibitors. So all along that dorsal mesoderm, you're getting BMP inhibitors. At the anterior side, you're also getting Wnt antagonists. And these Wnt antagonists are going to diffuse backwards. You know, as that thing's coming forward, it's leaving Wnt antagonists behind, but it's all coming from that anterior leading gastrulation structure. So you've got a gradient of Wnt blocking activity, high at the anterior side, low at the posterior side. And so what this is showing, this is slide 63, is that you can do this experimentally where first you take a animal cap, right? Take the animal cap, ectoderm, epidermal ectoderm. First you hit it with a BMP blocker to make it into neuroectoderm, and then you hit it with a certain concentration of Wnt blocking activity, okay? And you get another animal cap and you hit it with higher, another animal cap, hit it with higher. So you get a series of concentrations of Wnt blocking on these neurally induced animal caps. And what you find is at the highest Wnt blocking activity, you get anterior structures, anterior neural tissue. And at the lowest concentration with no Wnt, you're going to get the posterior neural tissue. Okay, so this gradient of BMP activity is giving you anterior to posterior axis. Okay. Questions about that? I feel like I've said this 100 times, but it gets repeated a lot in the book, and I know it's complicated, so it's worth repeating. Okay, and another thing that's important in this AP patterning is you also get FGF at the posterior end. If you remember from a few slides ago, I was talking about how you get FGF being produced here. And so once this gastrulates, you've got FGF still over here at the posterior end. And that also is going to decrease Wnt signaling and give you some posteriorization. I love words like posteriorization because if you were in an English class, you would lose points for it. But here in a biology class, you'll lose points for not using it. So that's why we're better than them. Mesoderm patterning. Okay. All right, so now we've talked about the anterior posterior of the neural tube, but if you go dorsal ventral in the mesoderm, we've also patterned that at the same time during neurulation. And that's kind of shown here where from anterior to posterior, you start getting this somite formation. So somites are laid down as a solid block of a solid kind of tube of tissue on either side of the nodal cord. And then once you get later into neurulation, that solid bit of tissue starts to block off as individual somites. And these are like the body segments, basically, going from anterior to posterior. And last but not least here, looking dorsal to ventral here, you've got everything you need to make an embryo at this point. You start out with epidermis, which is on top, but also covering the whole body. So this is that after neurulation. You got your neural tube, which is going to be brain and spinal cord, nodal cord, which induced that neural tube. And then on either side, you've got a somite, okay, left and right. So this somite, we'll see later, this will be really important once we get into mouse. The somite itself has got differentiation to it. Most of it, this yellow part, is sclerotome, and then this is dermotome. Not that important right now, but we'll see that the dermomyotome becomes the dermis, which is the layer right underneath your skin. Right under the epidermis is the dermis. So this part of the somite is going to become that, and the rest of this will become spinal cord. Your sclerotome will become spinal cord. And the nodal cord really important here for inducing that neural tube, but its importance gets less and less as development goes on, and eventually it ends up the spongy discs between the vertebrae. Okay, which doesn't seem like a big deal until you burst one. I broke one in my lower back once, way back in '99, and I had really bad sciatica because that little gushy thing, when it bursts out, it hits a nerve and puts pressure on the nerve and causes a lot of pain. That's neither here nor there. But okay, so you've got your somites, and then you've got this red stuff. So this is your lateral plate mesoderm. Okay, and the lateral plate mesoderm then will go on to form, your ribs, your axial skeleton, your shoulders and arms and legs and all of that. Okay, so all the body case is coming out of this. And then inside that body case is the gut. So this is your endoderm here, archenteron, which will be the gut lining, and then yolky endoderm until you use that yolk up, until the frog uses that yolk up, is down here. Okay. 138. All right, so that's what I got for today. Let me see your faces real quick. Whoever doesn't show their face is going to lose 5 bonus points. You can't lose bonus points if you don't have any. What am I talking about? That's a scare tactic. Okay, don't we feel more like a class when we do this? Hey, Rocky, you look bored. Good to see you, though. Okay, any questions about this? Comments? Discussion points? Any ethical implications we need to bring up? No? Okay, well, let's call it a day then. We'll meet back here again on Friday. And I'm not sure what we're going to do on Friday, but we will definitely start with a quiz. And then I'm debating then if we're going to go on to chapter 5 or finish chapter 4 and do zebrafish. I think we might do zebrafish, which is just a little different from the frog. Okay, guys. See you later.